Yeah. And hi, Michael. Sorry about all this. Good to see you. I just have a welcome you to VICC presentation, and it's my pleasure to welcome you and introduce you to all of you, Dr. Michael Kessler. He's current working at Regeneron Genetics Center, Regeneron Pharmaceutical Company, as a statistical geneticist, and I'm looking forward to your presentations.
Great. Thank you so much, Samanthi. Thanks for having me. Again, sorry to keep everyone. Glad we could find the solution. So my name is Michael Kessler. As mentioned, I work as a statistical geneticist at the Regeneron Genetics Center. And today I'll talk a little bit about work we've been doing with clonal hematopoiesis, how we can generate phenotypes across very large-scale exome sequencing data across multiple biobanks, and some genetic and phenotypic associations and translational follow-up we've been working on. I do want to just take this opportunity for a quick moment to note that I am an employee of Regeneron Pharmaceuticals and a stockholder. So just take this as a quick disclosure slide, and forgive the additional text at the bottom here. I was recently attending a translational medicine conference last week, and just going to talk about some similar work here today. Next slide, please. Okay, so just a quick overview of what I hope to touch on today. Actually, Samanthi, let me ask you, do you guys usually do this with questions in the middle or questions at the end?
I think with the time, we'll just would like to hear the whole slide. And of course, everybody is more informal discussion. So if there is a question, they can definitely ask, but it's
Sure.
like to go through the slides and your presentation also.
Sure, no problem. Sounds good. Okay, so I'll go over a little bit of background. I'm going to keep that light, just talk briefly about what clonal hematopoiesis is and get into more as we go through the slides. Talk about how we call clonal hematopoiesis in nearly 1 million people. The data I show you today will be from closer to 700,000 individuals. That reflects some of what we've been able to publish on recently. But we've extended this internally and are continuing to extend things further. I'm going to go through some genetic discovery, some targets of interest with some functional follow-up, and some additional phenotypic associations and genetic epidemiologic analyses. Next slide, please. Okay, so what is clonal hematopoiesis? So first off, I just want to make a clarifying distinction between what you might see as CHIP in the literature and CH. So CH is clonal hematopoiesis. It reflects the process by which cells in the blood compartment are derived from essentially clonal populations and start to take over an increasingly large percentage of the blood cells. This is more of an umbrella term. And the way I think about it, CHIP is a bit of a subsetted term. CHIP stands for clonal hematopoiesis of indeterminate potential and really is more of a, in my view, clinical term. It reflects individuals who have driver mutations identified in their blood and in blood cells with a variant allele fraction of greater than or equal to 2%. So that represents abasement as far as the number of cells that are carrying these markers that have expanded clonally, but they don't have other severe cytopenias or myeloproliferative neoplastic signs. And so they're considered individuals where there's pronounced and identifiable clonal hematopoiesis, but where it's unclear what the implications of this are. And that's sort of this CHIP term. So I'll use CH a little more throughout this talk, but I just wanted to clarify. So CH or CHIP is associated with aging. So you can see on the bottom right here from a nice review work from Juswal and Ebert, it's strongly associated with age. In fact, if you use error-corrected sequencing technologies which really probe into rarer and rarer somatic mutations, you can see what are the CHIP-like driver mutations or CHIP driver mutations in nearly everyone, depending on how deep you sequence. So this is just to emphasize that these mutations are there, that they're understood to be there, but at low levels, it's not exactly clear why they expand or at what rate they'll expand. But ultimately, when you use this more strict cutoff and you're looking at more expanded clonal hematopoietic events across people, you start to get to about a prevalence of estimated at about 10-15% by age 70. So with that backdrop and known associations with aging, with diseases such as cardiovascular disease and cancer, and even an association with overall mortality being increased in individuals who have this CHIP or, again, clonal hematopoietic expansions, we were interested in seeing what we could do with data on nearly 1 million people where we have at scale exome data. Next slide, please. So let's jump into the calling and what we did. So we first assembled information from the literature and leaning on some of the landmark papers in the field from Jaswal, from BIC, from PIC, and others. The general idea here is that with exome sequencing data, like we have roughly 30 to 50x coverage, very, very optimized, so minimal off-target reads. We had to really in our view, we were going to be able to only do this effectively if we leaned on what was known in the literature. So we narrowed in on about 50 or so genes. Ultimately, it was 56 genes that are known to be recurrently mutated in myeloid tissue, so in blood and in myeloid proliferative disease. And that allowed us to focus in on an array of filtering approaches, of heuristic filtering approaches that, in our view, gave us the best chance of generating a relatively clean call set that helped us, let's say, bring out the signal despite the challenge of calling these mutations from a backdrop of many germline variants and relatively low coverage. Next slide, please. So this is just a very high-level overview of the approach here. I'm happy to discuss it more if there are questions now or at the end. But essentially, we started using the MUTEC2 caller. It's a popular caller in the field. We used young people as a sort of control or you might consider it as a panel of normals, individuals who wouldn't be expected to have expanded somatic mutations. We used a lot of population-based data for other types of filtering. And to build up this heuristic approach I alluded to earlier, we, as I mentioned, subsetted to 56 known. It says CHIP genes, but these are recurrently mutated myeloid genes. We did an annotation and QC approach, this really kind of curated filtering strategy, which here I've labeled as these CHIP rules, but these are these sort of in-house rules we used based on known patterns in recurrently mutated CHIP genes about missense variants, indels, how consistent are their VAFs with being somatic across how many individuals in our data sets. And ultimately, we produced this final CHIP call set. So this was done, I just want to note, in collaboration with two of my colleagues at Regeneron, Amy Damask and Sean O'Keefe. Next slide, please. And ultimately, we generated this call set. So I'm not sure why this isn't rendering. Maybe it's... Actually, I have a quick question. Are you loading this off of Google Drive?
Yes.
Yeah, so it might, for me, at least on my end, when I do that, it leads to this blurry compression for some of the figures. I'm not sure if it's worth downloading and opening locally, but it may fix this if that's going to be the case in some of the other slides.
All right.
All right, I can keep going. I'm not sure how it'll affect the other ones, but
Yeah, give me a second. You can keep going and I'll try to download it in the meantime.
okay, thanks. Sounds good. So this is showing 23 of the 56 genes. They're blurry at the moment, but these are the most either the most common CHIP genes or a few towards the right end tail here where there's interest in the field, and we had some more confidence in the variant calls across those genes. But essentially, these represent about 95% of the calls found in the 56 genes. And so for a main analysis that we started with, we focused on these 23 genes. I'll just note this is work published last year by our group in Nature. If there's interest in additional details beyond what I can cover today, they're outlined with more depth in that work. This is the number of variants we called across these genes in both the UKB and GHS cohorts. This is the UK Biobank and Geisinger Health System, two of our largest cohorts. They comprise about 690,000 individuals represented in this work. And as I mentioned earlier, we've been working to extend this to additional cohorts beyond 6 at this point, cohorts that have increased ancestral representation as far as the individuals that are part of the biobank initiatives. And we're getting towards 1 million people that we're doing this somatic focused analysis on, in addition to other hematologic and oncologic analysis. Okay, so next slide, or perhaps local. Yeah, so hopefully we'll have a local option where some of these won't be as blurry, but I just wanted to show very quickly here. So the y-axis here represents the prevalence of CHIP. When we say prevalence, I'm taking all of the individuals who were carriers for any of the called driver mutations that I noted on the previous slide. So this is in this more narrow subset of 23 genes that are going to comprise the genes underpinning the analyses I'll be showing for the remaining figures. The prevalence is about 10 to 15% by age 70 in both cohorts, very consistent with what's been shown in the literature. And ultimately, just I'll note that the Geisinger health system has a wider age range. That's why we see data represented from below the age 20 up to 100, because in UKB, this was enrolled with strong focus on individuals between age 40 and 70. Okay, so we felt with some looking at these prevalence rates that this suggested the call set was reasonable and we were in the right ballpark here. Next slide, please. So I just want to note quickly here. So we started to think about what can we do with this call set. I think there's a number of things. One thing was trying to look for whether there are germline variants that are associated with your likelihood of having CHIP, that is having somatic expansion of blood cells due to driver mutations. So you can do that in the context of GWAS, GWAS XWAS. Previous efforts, as I'm showing here, were working with carrier numbers of CHIP carriers that were more than 10 times smaller than what we were able to ultimately assemble. For example, the most recent effort by VICC et al. Had nearly 4,000 CHIP carriers. We had over 40,000. So they were previously able to identify about 3 loci across these studies, replicating the signal at the TERT locus, which is one of the strongest signals as far as germline variants that associate with clonal hematopoiesis. Next slide, please. So we moved to extend this into what I had mentioned, or what I had called previously this genetic discovery effort through association testing. Next slide, please. And ultimately, we were able, this isn't rendering yet. I figured that may happen. So
Yeah,
I don't know
Google Chrome is just like choking out on downloading it. I'm going to try through Safari.
No problem. Maybe I'll stop for questions, Samanthi, just for a minute while we get the slides local so they'll render.
I think that's a great idea. If there is a question, please feel free to discussion for like a couple of minutes till Wes can download it. So I have one question. As we are going through the slides, I just realized that one of the things which you went very quickly is the variant calling and annotation. You said like one of the challenges we face in clinic is the coverage, and whole exome sequencing coverage you said is a 50x. So can you just elaborate that process? Like how do you call them and how do you kind of like curate them? Because these are considered to be a low variant allele frequency in the sequencing, right?
Sure. So happy to go through some more details. So just to make sure I'm thinking about it as similarly, you mean like 50x would be considered pretty low from a clinical perspective? Is that correct?
Yes. So
Yeah.
How do you get the confidence about that, especially for the cancer patients where you do have a P53 and circulating DNA also kind of there?
Yeah, absolutely. So a number of things here. It's such an important point, and I appreciate you asking. So first of all, let me work backwards with my answer. So first of all, I totally agree about TP53 in particular. I think it's one of the ones that's harder to separate, and probably one of the ones where we'll have the most challenge here, even longer term. We've talked quite a bit about TP53 in this context internally as well. But one thing we've done more recently is we've developed like a chip panel, which has coverages in the range of 300x, specifically with the goal of improving how we can call variants, particularly somatic variants, across these notably recurrent genes and the regions that feature these genes, notably recurrently mutated, I should say, myeloid genes. So that as a part of that process, we've done comparisons. We actually have like a callback study we're working on with certain individuals that have CHIP, where we can follow them over time, and controls. And we've done comparison with the kind of CHIP capture panel that we've updated versus the original population scale, lower coverage, sort of optimized exome, and we're seeing really good concordance. So like I mentioned, I'll go backwards here with this explanation. So we think by focusing in on myeloid, recurrently mutated myeloid genes, what's known about some of the specific hotspot mutations, and some of the heuristics I'll describe in just a moment, we're able to still do reasonably well with 50x. So what we essentially do is we start with this blind, if you will, or unbiased, let's say, somatic mutation calling. And we're just using a standard caller. And in our view, the initial calls that come out are very noisy. They're really just they're very, very noisy. And ultimately, we have about 15 or 20 steps where we refine things down, right? So first of all, we'll require a minimum depth consistent with the alternate allele. I think we usually use 3. We can call these down to about 0.5%. If 0.5% of the total depth is comprised of alternate allele reads, we think we do okay, but for the analysis, we've been more strict to 2%. So we're just increasing the likelihood of having more support for the call. We do a lot of population-based filtering. In other words, for example, just to give you one example, if you take like recurrent JAK2 missense variants or some of the most common hotspot DNMT3A or ASXL1 variants, those should represent the most recurrent variants. And so if we start to see other variants that have larger effects in very large population databases like NOMAD, or even in our internal data where we have another, let's say 500,000 individuals roughly, we can filter those out as likely to actually be rare germline variants where we're just calling them. Maybe there's enough people that have variant allele fraction skews. We're doing some simple statistical tests using binomial sampling, expectations, certain mutations we know that have a lot of indels where we have to be particularly concerned. We're using Mutex panel of normals flag across all of our cohorts. So we sort of take a union and say, if we're worried you're a normal or germline in any cohort, we're going to filter you from all cohorts. And then I'll just say, finally, we rely quite heavily on efforts by Sid Jaswal, Ben Ebert's lab, Alex Bick's work, that have done a lot of sort of manual curation for variants. There's even some of them that have been followed up functionally, and we rely heavily on those. So with a few exceptions where we're strict on our filters, we're permissive to some of the variants that have been repeatedly called as likely to be somatic chip variants. So I don't know if that, I know we have the slides back now, but Sumanthi, I don't know if that gets at the full scope of your question, but hopefully adds a little bit of detail and clarity.
Yeah, thank you. Yes.
Sure.
And chip panel is on 300x sounds a very good approach also to kind of like follow up for a larger, for especially more the clinical. Yes, thank you. Okay, so we got your slide.
Yep, absolutely. So thank you guys. Okay, so with regard to this genetic discovery, we were able to identify numerous novel common variant loci that are associated with being a chip carrier. Again, this is done with a binary coding of being a chip carrier. It's combining all individuals that have recurrent driver mutations that we identified in any of the 23 chip genes. We recapitulate previously known association signals as seen with the gene names that are colored in black. There are a few in gray that are sort of subthreshold, but I'll get to them in the next figure. And ultimately, there's 21 signals that are novel where we're putting the nearest gene. So we thought this was pretty encouraging. A lot of these are related to, these are either genes known to be related to myeloid biology, or other elements of DNA repair, myeloid differentiation, or really just hematopoiesis in general. Next slide, please. Actually, sorry, can you go back one slide? I'll just mention quickly. So this is done in UKB alone, and we used GHS in this effort for replication. So we replicate about 75% of the signals where there's power. We've ultimately extended this, as I mentioned, to numerous additional cohorts, and we're doing analysis now where we're meta-analyzing these signals. And I'm not going to present it today, but hopefully we'll be able to make that public over the intermediate term. We're able to see numerous additional loci as we increase our power. So really tending towards about 50 loci, adding in some additional genes. So the sample sizes that we're seeing now really are reflecting 50, 60,000 chip carriers and hundreds of thousands, well over 500,000 controls. Okay, thanks. Next slide, please. So here I'm showing Manhattan plots again, but we're breaking down the phenotype used in these association analyses into chip subtypes. So for example, in the top panel, you'd be considered a chip carrier if you had DNMT3A chip. So that is recurrent variants called as somatic in recurrently mutated chip genes, in this case DNMT3A. You'd be considered a carrier here only if you have driver mutations in TET2 that we've called ASXL1, et cetera. And we did this in about 6 or 7 of the most recurrent chip subtypes. I'm only showing the top 3 here, just to emphasize the point that some of these loci are shared across chip subtypes. So for example, TERT, or even what you see here at ATM. And yes, I just want to note, we have smaller sample size, for example, with ASXL1, so there's less power, but we've looked at effect size estimates as well. And these patterns of shared, unique, or opposing germline effects hold. For example, a unique effect seems to be the signal at the THRB locus, which is specific to TET2. And perhaps most notably is this signal seen at TCL1A. So this has been published on kind of over the last year or two as well by some other groups. We were able to show this with really convincing support, statistically speaking. Others have done some really nice work, particularly Weinstock et al., to do some functional follow-up and suggest that this really is an interaction in non-DNMT3A chip subtypes. So just to walk through this quickly, carriers of these common variants are at about a 15-20% elevated risk of being DNMT3A chip carriers. But conversely, they're at about a 20% reduced risk of being chip carriers of other chip subtypes. Here, I'm just showing TET2 and ASXL1. So there's a question as to whether if you're at a reduced risk of developing TET2 or ASXL1 chip due to what it seems from, as I mentioned, the recent functional work seems to be reduction in TCL1A expression or function, and a dependence among these non-DNMT3A chip subtypes on the function of TCL1A, and actually increased expression as these start to clonally expand after acquiring these mutations. Perhaps your hematopoietic compartment is more susceptible to DNMT3A expansion, and that's why you see an increased risk, whereas it's not necessarily related to some additional interaction effect biologically between TCL1A and DNMT3A. That seems to be what some of this recent work that's just come out over the last 6 months or so suggests. But nonetheless, it's very interesting that this signal is completely gone in the previous figure. There is no signal at the TCL1A locus, but it ends up being that it's actually a very strong signal when you decompose this into the chip subtypes that are aggregated in that overall chip phenotype and overall chip analysis. Next slide, please. Excuse me. Okay, so I want to focus on 2 specific signals here. I think they're pretty interesting, and perhaps most notably they represent a nice example of what we can do translationally, where we're taking this human genetics first approach to identifying signals, to working with phenotypes at scale that previously were captured and considered in many fewer individuals, and ultimately further explore them biologically and using experimental follow-up in order to do basic biology, as well as translate these findings, hopefully over time, towards therapeutic goals. Next slide, please. So just to reiterate, this is showing the results from our overall chip analysis and from our DNMT3A analysis. You see this signal in LY75 when we look just at the DNMT3A analysis, the particular index SNP, the signal that has the variant, I should say, with the strongest signal actually shows risk, is associated with risk reduction. Next slide, please. And it seems to be that this signal is specific to DNMT3A. So we ended up performing statistical fine mapping, which uses linkage information and haplotypic information to do a variable selection procedure and attempt to estimate which variants are most likely to be driving signals across this locus. So at the top, we're looking at this locus zoom plot where we're just kind of looking at the linkage disequilibrium across this region, as well as the p-value, which here is shown in -log10 transformation on the y-axis of the variants in this area. And I'm highlighting this one particular variant that doesn't seem like it's a leading signal or any signal that would, any variant that would be driving signal here. But when you look at the CPIP, just stands for conditional posterior probability, but conditional posterior inclusion probability. But the main point here is that it's estimating how many credible sets are most parsimonious statistically to be found at this locus. That is to say, you can think of it as clusters or a K choice. Like what's the most parsimonious configuration here of signals based on the correlation structure of the haplotypes? And it turns out that the answer in this case is 3. There's most likely to be 3. And for one of them, this rare variant is highly likely to be the variant that drives that association signal. You can kind of identify this. We think it's a really nice example where statistical fine mapping can give you a lot more confidence about what's likely to be actually a causal variant when doing association analyses. And given that this is a missense variant, we obviously have some prior biological expectation that it may in fact be functional. Next slide, please. So we can actually take this a bit further. So here we're doing this based on the statistical output, association output from our DNMT3A chip specific analysis. And we're extending the haplotypes and the minor allele frequency cutoff of the variants that we're including in this modeling procedure. And actually, we see that the most parsimonious number of independent signals is 4, and another one of the variants that's leading the credible set, that's over 50% likely to be driving it, in fact, is another missense variant shown by this RSID here, which is actually 10 times rarer than that other relatively, I was calling it rare common variant. So that one was at about 2%. This one's at 0.2% roughly. Next slide, please. And if we just look at these in some forest plots with tables here, you can see these are the association signals here and the estimated effects here are consistent across cohorts. When you meta-analyze, we have pretty, in our view, compelling statistical support for these signals. The variant that's found at about 2% reduces your risk of having DNMT3A chip by over 20%. Variant that's 10 times rarer reduces your risk by about 50%. Both of these are missense variants. They are predicted by CAD to be damaging. So we thought this was really interesting and suggests that some kind of potentially disruption of LY75, this lymphocytic protein, somehow reduces the likelihood of being a DNMT3A specific chip carrier. And we have both independent and rare variant signals. So these are, in our view, this is a bit of orthogonal evidence in some genetic sense. These are not linked. They're across the site frequency spectrum. We thought this was quite interesting. Next slide, please. So I know we're a little short on time, so I'm gonna pick up the pace a bit, but this is just very quick background on LY75. It's a lymphocytic antigen, as I mentioned. It's thought to be involved in antigen processing, presentation. It's predominantly found in dendritic cells, in particular migratory dendritic cells. When you ablate it, experiments have shown that it impairs T cell homeostasis and various aspects of proliferation and function. Next slide, please. So I'm just showing one slide here, and then we're going to jump to another signal we discovered and some additional translational follow-up. But I just wanted to show a little bit here of some more recent work that we've done. So this is, in quotes, a little bit more hot off the press. But I just want to show it as an example of how we're thinking about this and working to try and translate some of these signals that we identified in this genetics first approach using this at scale human genetics data. So, but what I'm showing here just very briefly is what happens when you do LY75 knockouts using genetic engineering approaches. And this is done in collaboration with a colleague of mine, Michael Van Meter. And ultimately, if you look at cells that are wild type for LY75, you look how they bind this CpG-ODN ligand, this ligand of LY75, you see that it binds, notably, if you knock it out, the binding is gone. But ultimately, if you knock these LY75 back in using constructs where these mutations have been knocked in, you don't recapitulate the binding that you see with wild type. In fact, there's disrupted binding. Again, this is just a first step. There, I think, in my view and in our view, like a lot of fun, exciting science, this probably opens more questions up than it answers. But it does seem to suggest that these variants are disrupting ligand binding. That's a little. That was interesting to us because these aren't in canonical binding regions of the LY75 protein. So that's sort of now an open question. We're going to extend this a bit further, add some positive and negative, some additional positive negative controls. And really the bigger question in our view is, okay, let's assume for a second that this is affecting binding. We actually want to first test if it's just directly affecting expression or transport of LY75 rather than direct binding. So that's something that's kind of ongoing right now. And then perhaps a bigger question is, why would LY75, this typical sort of lymphocytic protein you might think would be more traditionally related to lymphoid cell development, why would it have this effect, this what seems from the genetics data to be this notable effect on reducing your risk of DNMT3A expanded clonal hematopoiesis? So that remains really an open question, but we're taking this genetics first approach, as I mentioned, and trusting that. And so we're working now to further disentangle this. Next, let me stop for just a second. Are there any questions? All right, next slide.
Michael, I do have a question. Can you hear me?
Yes.
So, how did you treat all the variants you found in terms of oncogenicity classification, figuring out if those are drivers or passenger mutations? What was your approach? Like, for instance, DNMT3A and TET2, we have many missense variations that are not well characterized. And how did you treat that?
Sure. So many of the ones that the ones that make up what I'll call the majority of our call set, we were really heavily relying on the literature. So ones that had previously been published across studies of either clonal hematopoiesis itself or myeloid neoplastic from data generated from individuals with myeloid neoplasias. But we kind of, to your point, trusted the biological annotations and otherwise our heuristic filtering approach, right? So, was the variant allele fraction consistently low across all individuals who had that variant? If it occurs, once, there's only so much we can do. So we wanted to know, is it a missense variant? What is how has it been seen or not seen in external and internal data that are germline call sets, right? So another element here is with some of the newest call sets with Google's deep variant, not always, but these germline call sets are actually very, very clean. So if this is seen as germline before, if there's some people that have that in a germline call set where the variant allele fraction is really consistent with expectation of being germline, we were going to filter them out, right? So the biggest, I would say, component of our heuristic filtering, and there's like I mentioned about 20. So, I could dig in further. I have some tables in backup with some additional aspects. For example, we focused on homopolymer repeats where they would reduce we would filter more out if there was evidence that they were within homopolymer regions of the genome. There were other elements as far as if they were changes that are consistent with oxidative damage. We required higher read counts. So we used a lot of that heuristic filtering. But to your point, if all we knew was that it's a missense and it's passing all of our filters, to us that it was then seen as consistent enough with somatic. Again, the point being seen in our relatively low coverage data with enough reads representing what seems like enough expansion that it's marking CHIP. And to your specific question about driver versus passenger, even if it's a passenger variant, our view essentially was that's fairly parsimonious with still being someone with clonal hematopoiesis. It might add a little bit of noise whether it's actually the gene it's found in, but we think it'll be quite rare where you have clonal hematopoiesis driven by another CHIP mutation that we're not seeing. I should mention in this whole analysis, we excluded anyone who had in the CHIP subtype specific analyses, we exclude anyone who has any multi-hit CHIP. That is to say, if you had any driver mutations or expanded somatic mutations in more than one CHIP gene, you were only included in the overall CHIP analysis and not the CHIP subtype analyses. So that should limit some of that confounding in our view as well. Does that answer what you were asking to a certain degree?
Yes, no, I think that's very interesting. So basically, you didn't differentiate passengers from drivers, right? If the variant was there, you assume, and it was a little frequency higher than your threshold, you assume that was a clonal hematopoiesis and caused by that variant. Yeah, yeah, it's really hard. I mean, to figure that out, right? Mainly
Absolutely.
with that big amount of data. But okay,
Yeah,
yes, that
absolutely.
answers.
No, absolutely. I totally agree with you. And actually, to the point Samanthi made earlier that we were chatting about briefly, we were really wondering this when we did this callback study. And as we extended these panels and some of our new data, we've changed some of the exome capturing technology as well, like at scale on, let's say, the newest 500,000 samples we've sequenced. And we were really pleasantly, we were quite, we felt good about the fact that we were able to replicate things really well. We were replicating our call sets at about 98, 99%. That is to say, about 99% of individuals that we had called as chip carriers were chip carriers when we used the deeper 300x sequencing approach. Okay, next slide, please. So I know we're short on time here. So, what I don't know, Samanthi, can I ask you, like, is there probably more interest in another target with some more functional work, or getting into some of the phenotypic associations and that piece here for the last few minutes?
I think we'll just go with the phenotypic association.
Okay, so let's skip over this. We can click through the next handful of slides. The quick takeaway is we did some functional work and actually found a direct interaction between PARP and DNMT3A. But I can get into that maybe in some future work or a future event. Okay, so looking at phenotypes now. So we decided to do a phenome-wide analysis that is taking the chip carrier statuses that I've described previously and running association analyses with thousands of traits. Next slide, please. So this is really just a quick snapshot of what does the phenotypic association landscape look like across chip, essentially, for individuals of chip that we called as being driven by mutations in these various genes. There's a quick couple of overall takeaways, which would be very fast. It's predominantly associated with hematologic, cardiovascular, and oncologic traits, just like one might expect from the literature. In certain things like ASXL1, where you see respiratory associations, these are largely driven by smoking. DNMT3A seems to be associated with cancer more so than others, JAK2 with cardiovascular disease, and certain hematologic traits, quantitative traits. SUS12 had this kind of atypical profile, but it's quite rare. We use these FERT stabilized association models that are regularized and fairly stable for rare counts, but still with rare counts, the confidence is lower. But we did find this a bit interesting and are keeping our eye on it as we scale our data. Next slide, please. So one thing we first wanted to look at was at associations between chip and cardiovascular disease. These have been described numerous times in the literature. Odds ratios of 2, 3, 4x risk of cardiovascular disease amongst chip carriers have been reported. We did not find this to be the case. We first off found that in the first 50K samples that were published on from the UKB, the association seems to be stronger. There seems to be an ascertainment effect here where we find overall only about a 10% increased risk. You can see that here as well. We don't find much signal as well in GHS either. We do find the signal is there, which is modest, is more pronounced for TET2, and we in our view think this is real. There is an effect that's in our view a bit more modest than previously described. But there's of course questions about variant filtering and such. We've done this. We've done sensitivity analyses here to do it many different ways with increasing strictness. We generally find the effects are not the 2, 3, 4-fold risk that we've seen. And again, because we're using genetics here, we think these are well calibrated to be robust to bidirectional confounding. Next slide, please. So yeah, so we skipped the slide before, but the previous slide shows a result reported by VICC et al. That if you have chip, but also have this missense mutation in IL-6R, that is thought to be sort of a genetic proxy for reduced IL-6R function by knocking out some function, you're actually resistant to cardiovascular disease. There's much less cardiovascular disease. Next slide, please. We did not find this to be the case when expanding to the 450K. We are able to reproduce it in the 50K. When we expand to the 450K, we see no signal. Let me just note, this also includes the 50K. If we just go to the 400K, there really is no signal at all. So we think this is likely to have been some kind of winner's curse, or maybe there's ascertainment bias in these 50K, maybe they ascertained on some cardiac imaging or potentially cardiac based pathology. Next slide, please. And so here I'm just summarizing. We also did a lot of work with polygenic risk scores and particularly Mendelian randomization to basically ask, hey, if we can use the 50 or so independent signals we identified as germline variants associated with the likelihood of somatic expansion, can we see how those variants associate with other diseases as a way to try and use a genetic epidemiologic approach to really ascertain which diseases really are associated in causal fashion with CHIP, right? Because now it's not you have CHIP, you have another disease, it's your likelihood of having CHIP based on the germline genetics you're born with. And ultimately, we acknowledge up front, we don't have really great power here. This isn't the most heritable trait. These variants only explain around 2% of the variation in CHIP. And also, generally some of these GWAS are underpowered across traits, but it's at least a start, and it can give us evidence of association when we see it in our view. And what we do see is, first, we do see strong, compelling associations with CHIP in our validation cohort, that is the Geisinger health system. So if you have variants that are associating with CHIP and UKB, we can use Mendelian randomization to show strong associations with your likelihood of being a CHIP carrier in GHS. We also find strong associations with a few cancers, namely blood cancers as expected, skin cancers, both melanoma and non-melanoma, and lung cancer. And we found this quite interesting, especially because the cancers here, other than blood cancer itself, are those that are most responsive to immune checkpoint therapy. So we started wondering about whether there's some immune system by solid tumor effect here that may relate to CHIP, or perhaps even be mediated by CHIP. Are individuals that have clonal hematopoiesis having some effect on immunosurveillance, immunodeficiency, some inflammatory effect. Next slide, please. I know we're running out of time here, so I'll just wrap down with another. I think this is the last slide. So again, one slide of showing results from some experimental work that follows this up. Again, this is really kind of work we've just done over the last few weeks and months. We're extending this further, but my colleague Mike Van Meter and our group have worked together to try and develop some mouse models that can test this more directly. So there's actually two models here. One is a full knockout, heterozygous knockout of TET2. One is a transplantation of bone marrow that's been knocked out for TET2, so trying to control what tissues have TET2 knockout. But in both cases, you see that when you transplant lung cells, you see significantly increased tumor growth over time in the mice that have TET2 knockout in the heterozygous form in their bone marrow. So this is trying to recapitulate CHIP and test what's the effect on solid tumor formation. We didn't necessarily expect this, but we think it's quite interesting. There's a lot of work to do to follow this up. Why is this the case? What is the mechanism? Is it seen in other cancer models? But we thought it was really interesting. There's some other literature. Next slide, please. I'm not sure if I added it here or to the background. No, I have it in the background, but I'll just say briefly, there's some other work showing that CHIP mutations that have been convincingly traced to the myeloid cell lineages have been found in the tumor microenvironment. Other work suggesting that, in fact, CHIP carriers may be at an accelerated risk directly in experimental context of solid tumor formation. So we think that's really interesting and just represents another example of how we can move things in a translational way forward, start to learn basic biology, start to interface between hematology, oncology, and immunology by taking this genetics first approach at scale. Yeah, so next slide, please. I'll try to just think I have an acknowledgements and just thank everyone. We can skip the summary, but next slide, please. Yeah, so just want to acknowledge Regeneron, the Regeneron Genetics Center, the cohorts we worked with, and some of the additional ones that we're working with, but haven't shown data from those efforts here today. Many of my colleagues, great colleagues. I'm very, very appreciative to work with them. And with that, I think if you push the arrow, there's some questions, and I think we're out of time, but I just want to thank everyone. Sorry again for the technical difficulties up front.
Thank you, Michael, for the very large amount of data explanation. And I know this is so many things packed together, the pipeline, the curation, and also the biology. So fantastic work. I'm very impressed. So I know with the time we are over the time. So I just want to just give chance for people who would like to stay, and just maybe a couple of minutes we can spend on questions. If there is a question, please. Otherwise, thank you so much for everybody, and sorry about the technical issues. So if you have any question, please go ahead.
Yeah. And thanks again to everyone for having me. I tried to read a little bit about the group. It seems really, really interesting, super cool. So thank you. Appreciate it.
Bye. Hi, Michael. This is Dr.
Hi, sorry, I'm not sure if we can, it's cutting out for me, but
Yeah,
if you
we're
have,
not.
I'm happy to take any questions. Yeah.
Hi, Michael. Can you hear me?
Your connection is pretty bad.
Hello, Michael. Can you hear me?
Yes, I can now. Yes.
Okay. Okay. So I will try to. So. Oh, okay. I will post it in the group. No problem. I will post my question in the group.
Okay. Sounds good. If anyone has any other questions, happy to try to answer any.
I think we should be good. Thank you once again for everybody for joining us, and also Michael. And have a good luck, and hopefully this is we'll see you in ASHG, American Society of Human Genetics, in a couple of
So Monty,
weeks from now.
we did have the question come through in chat.
Oh, okay. Oh, sorry. So maybe Wes, you can go ahead and ask the questions.
Yeah, the question was, could you please explain how your criteria for defining CH versus CHIP versus CHOP?
Sure. So can you actually, I'm not familiar with CHOP. Can you just clarify CHOP?
Yeah. So CHOP is nothing but the next level of the chip. So the chip is clonal hematopoiesis with intermittent potential, whereas the CHOP is clonal hematopoiesis with oncogenic potential. So only we are confirming this is a chip. So when we are confirming as a CHOP, that's where the clinical implication is coming into the picture. So is there any criteria you have been defining over here?
Yeah. So really good question. So the short answer is no, but let me give a little bit of context. So we use the term, maybe it's the same, maybe there's more clinical nuance I'm not familiar with, but we use the terms chip, so CH, chip, and then CCUS, right? So in our kind of the way we've been thinking about it, CH is really broad. So it would also include other evidence of any kind of clonal hematopoiesis. And the way we've thought about it, genetics first, is other signs of mutational markers representing clonal expansion. So things like mosaic loss of Y, mosaic loss of X, even perhaps lymphoid recurrent mutations, things that we're thinking about and working on and trying to develop further. If you meet what we're considering the clinical definition of CHIP, so you'd have 2% variant allele fraction, you would not have cytopenias. If you started to have cytopenias, we would think of you as CCUS, or clonal cytopenia of unknown significance. And we're thinking about that from like an angle of progression. For the analyses I've shown today and a lot of the at-scale analyses, we are not yet considering these additional lines of evidence for what you might consider progression, like quantitative evidence of cytopenias from quantitative blood traits. That is something we're interested in doing, particularly for various types of longitudinal modeling. But right now, the real goal was to ask, if you have a genetically defined marker of clonal hematopoietic expansion, particularly in this case, in the myeloid sense, do we find evidence of germline predictors for that somatic presentation? And in our view, it's not important how far down you are yet. I will say that in all the analyses I showed, we excluded individuals who had full-blown myeloid disease prior to their enrollment in the biobanks and collection of their blood samples, because we just think that really affects the sequencing, the confidence of calls, et cetera. So we excluded a few thousand people. We think that made some of these conservative. Yeah, but does that answer your question?
Yeah, so I got the pretty much little bit of idea like where you are coming from. So thanks for explaining this, Michael. And I have one more quick question. So have you connected this clonal hematopoiesis with the novel mutations also with the efficacy in solid tumor, especially in the immunotherapy efficacy? So is there any of your marker is affecting the immunotherapy efficacy?
Yeah, it's a fantastic question. I will say we're very interested in that. It's something we're looking into and trying to think about further, right? We have we've been working with some of our clinical trial teams, and some of them directly interested in this question, some interested in connected questions. So the answer I'll tell you is it's not clear to us yet, but it's really of interest. I totally agree with the importance of the question. Yeah.
Thanks, Michael. Similar line of thoughts also I'm working on. Basically, what I identified is that basically first I am a clinical genomist. So what I understand from foundation medicines, whenever I'm working with a medical oncologist, the liquid biopsy when they're done, particular gene of clonal hematopoiesis is getting higher version. When it is having higher version, the immunotherapy is not working for the patient. So that means it's already compromised the immunotherapy efficacy, that particular mutations and all. So that's what a
Absolutely.
little bit interesting. Thanks for your sharing your thoughts. Definitely it is going to be helpful for me also. Yeah.
No, absolutely. Totally agree. And
Thank
yeah,
you.
thank you for the great question. So I don't know, I'm happy to stay another minute or two if there's other questions, but whatever Sumanti is preferred. And
Wes, do we have any more questions? If not, then it's
No.
already. Okay, so thank you once again and see you. And thank you all for your time.
Thank you.
absolutely, thank you.